Yeah, so today I'll be talking about accelerating biomedical discovery with large-scale knowledge assembly and human-machine collaboration. And I work at the Lab of Systems Pharmacology at Harvard Medical School, and the mission of the lab is to reinvent the science of drug discovery by going beyond just understanding the direct targets of drugs and trying to understand the complex interconnected process by which cells react to perturbations, and through this be able to design novel therapies and understand existing ones and in which context they might work. And so the classic target-centric view of drugs is that you perturb a pretty much linear pathway within a cell with a drug that has a single well-defined target to modulate a single well-defined phenotype. And using this approach, you can assign therapies to different groups of patients. But the reality is much more like this diagram on the right-hand side, where the cell's biochemical pathways are heavily interconnected with all sorts of feedbacks and crosstalks. Drugs typically don't have a single well-defined target, but target different proteins in different pathways to different extents. There are various compensatory effects. There are multiple phenotypes that one has to take into account. And things like the cell's microenvironment also play an important role in how it responds and adapts to perturbations. And so computational modeling can crucially help in understanding this complexity and making decisions in the face of this knowledge about biochemical processes and mechanisms. However, in the field of systems biology that often deals with modeling these systems, there seems to be a disconnect where part of the community focuses on so-called bottom-up modeling, that is detailed mechanistic studies focusing on a relatively small subset of biochemical entities and creating detailed mathematical models of these pathways that have the potential to model specific doses and time courses and detailed mechanistic interactions. On the other hand, many studies, especially ones that work with large scale, often genome scale, high throughput data, they resort to statistical approaches such as gene set enrichment or simple approaches to dimensionality reduction to generate hypotheses without taking into account all the available knowledge about biochemical mechanisms. And so our goal is to try to draw on machine assembly and machine assistance to be able to bring detailed models to a larger scale, to be able to connect them to the scale of high throughput data that is routinely collected in labs nowadays. And so whether a human or a machine is doing modeling, this is one way to conceptualize the modeling process. That is, first we collect information from knowledge sources, we assemble or synthesize that knowledge, and then encode it in a mathematical form to create a model, while also taking into account context-specific data to, for instance, calibrate or contextualize that model. And ultimately, models help us construct explanations to what we have observed and predict outcomes under yet unobserved conditions. And so focusing specifically on machine-assisted modeling, we can draw on knowledge sources like pathway databases, the scientific literature, which is of course an unstructured encoding of knowledge, and expert input, and then have a layer of knowledge assembly that more specifically comprises of a knowledge representation, that is a standardized representation of pieces of knowledge from our sources. And this knowledge assembly layer also has to perform various operations to bring this knowledge into a form where it can be usefully used for modeling. So for instance, one has to correct for certain systematic errors, such as text mining errors coming from these sources, resolve redundancies among pieces of knowledge in a principled way, assess relevance to a given use case, assess believability of source knowledge, and infer missing information when something isn't explicitly mentioned in the sources but can be inferred from various pieces that are available. So we implemented this system called INDRA, the Integrated Network and Dynamical Reasoning Assembler, which is basically a software implementation of the same automated modeling concept as this diagram before. So on the left-hand side, we have modules for knowledge processing from different sources. These include APIs and processors to pathway databases, and also input APIs for text mining systems, which can read scientific publications. And all of these sources produce standardized INDRA statements, which are the central representation of INDRA for knowledge that it collects. And all these red nodes here, they are internal knowledge assembly processes that operate over INDRA statements to perform various assembly tasks. And once we have a coherent set of INDRA statements, we can generate multiple different types of models and outputs from the same underlying knowledge. And these include, for instance, generating rule-based dynamical models using the PySB framework, generating different kinds of network representations for the purpose of interactive visualization or causal path analysis. So Cytoscape.js is a commonly used web-based display approach for networks. SVGN is a widely used standard, again, for interactive graphical interaction. We can also generate HTML pages that allow browsing and curating statements and their underlying evidence. And we can even assemble into English language paragraphs, for instance, to, in a textual form, relay what INDRA has learned from certain sources. And just to emphasize here, INDRA actually has a lot more inputs than what is shown here, about 30 plus input APIs at this point, and 14 different output assemblers. So focusing on inputs first. So as I mentioned, INDRA takes both pathway databases and other structured sources such as drug target interaction databases as input. Here's a table that shows the current set of input sources. And one might think that information coming from these sources is kind of well-behaved and high quality. And it is high quality in the sense that these are human curated resources, but it still poses a lot of challenges to integrate this information consistently. And here is just one example coming from the Pathway Commons database, which itself draws on multiple curated resources and represents them in a format called BioPEX. These are biochemical reaction representations in Pathway Commons for the reaction by which MAC phosphorylates ERK. So MAP2K1 and MAPK1 are the two entities here. And you can see how many different implementations of this same knowledge there are. So for instance, these two reactions, they don't have any state given to the MAP2K1 protein, and they provide a single phosphorylation site on MAPK1 in two separate reactions. This is curated in the NetPath database that Pathway Commons integrates. However, for instance, in Phosphosite Plus, there is a single control structure that encodes both of these reactions for the two phosphorylation sites that are being set on MAPK1. In yet other sources, MAP2K1 also has some state, so it itself is phosphorylated and shown to be active, and ERK becomes active when it's phosphorylated. This is not curated in these sources, and so on and so forth. So even in these human curated resources, there's a big challenge of, again, knowledge assembly. On the other hand, we understand that most biomedical knowledge is actually residing in the scientific literature, and only a small subset has been curated into pathway databases. And the curation of pathway databases is actually a very expensive and resource-intensive process and requires a lot of human labor. And so it's tempting to think of processing the biomedical literature directly to derive some known and reported mechanisms. And so if you look at some examples of what papers might contain, very often we find that papers contain just one fragment of a much larger puzzle, such as this paper specifically discusses the negative regulation of BRAF by AKT. This paper talks about how ERK phosphorylation can lead to ALK1 activation, and this paper describes the effects of FGF1 on EGFR and VEGFR signaling. And so one might imagine extracting mechanisms described in each of these papers and then putting them together to put the pieces of the puzzle back together that are by nature fragmented in millions upon millions of publications. And there is, in fact, about 1.4 million new papers that appear every year on PubMed. Not all of them are at the molecular level, of course, but still, that's roughly the scale of these publications, about 4,000 new papers a day. And according to one estimate, approximately 20% of those papers are relevant to cancer biology. So about hundreds of papers a day. So natural language processing systems can take sentences such as the example here, like ASPP2 can be phosphorylated at serine 827 by MAPK1 and produce a machine-readable formal interpretation or representation of this unstructured knowledge. And here's an example from the TRIPS system developed by our collaborators at IHMC. And you can see how this sentence was turned into a semantic interpretation graph with a node for phosphorylation, and it has well-defined arguments, an affected protein, which is ASPP2, and an agent, controller MAPK1, and the site of phosphorylation is also extracted as serine 827, along with some other things like modality and TENS and so on. Another example of a machine reading system, the REACH system, developed by our collaborators at the University of Arizona. It uses a syntactic parse of the underlying text, like the sentence here that says that RAS resulted in upregulation of the MKP3 gene, and then uses these human-engineered graph patterns on that dependency graph to extract patterns of linguistic patterns of how different regulations and interactions are described in text. INDRA is actually integrated with several further systems. They each are engineered in different ways. They have different properties. They usually contain some components of machine learning. Some of them are fully machine learned, but very often these systems combine machine learned components with some explicit human engineering. And so these systems that are listed here are ones that are biology specific. But INDRA is actually also integrated with some general purpose causal relation extraction systems that aren't specific to biology. And so once we collect a number of inputs from text mining and pathway databases, we are faced with this challenge of somehow consistently assembling these pieces, somewhat similar to genome assembly from fragments of sequences. So you can imagine different statements coming in from sources that look like these boxes here. Again, taking the example of MAC phosphorylating ERK. Some of them are exact duplicates, like these three here. Some of them have more detail than others, but effectively describe the same mechanism. Some of them have errors, like an amino acid site here is incorrectly annotated, a named entity is incorrectly recognized. So instead of a protein family, it's recognized as a chemical here. The subject and the object are accidentally flipped due to a text mining error. And somehow at the end of the day, we have to end up with a single consistent representation of this mechanism, ideally with as much detail as can be reliably inferred, to end up with assembled mechanisms from these kind of raw extractions. So the underlying representation that we use in INDRA is a set of statement objects. Here's a diagram of these statement objects so you can get an overview. So a statement would be something like phosphorylation, which has an enzyme and a substrate, or activation, or amount regulation to represent, for instance, transcriptional regulation relationships. And the arguments of these statements are agents that can represent proteins, small molecules, biological processes, etc. Especially protein agents can have various kinds of state conditions. So we can represent post-translational modifications, activities, bound conditions, and so on. And each statement, importantly, also carries one or more pieces of evidence that actually link it back to where it was extracted from a particular database, a particular paper and sentence with further annotations. So one of the key tasks in this process is to fix systematic errors coming from sources. And one very common example of that is mistakes in named entity recognition, for instance, recognizing the synonym of a protein and assigning an identifier to it. So as one such example, in INDRA, we implemented a machine learned process for correcting for the resolution of ambiguous named entities. One example is ER, which can stand for many different things. So ER, when you see it in a publication, it can stand for endoplasmic reticulum, it can stand for estrogen receptor, it can also represent the emergency room, for instance. So for several such ambiguous strings, we built models that can predict based on additional text context, like the publication's text that this string appeared in, to predict the likely sense in which it was mentioned. And so in this example, for instance, there's a sentence that says stress was observed in several cellular components, including ER. The likely resolution of this is endoplasmic reticulum, whereas in this sentence, this has been observed in many types of breast cancer, including triple negative and ER/PR positive. It's much more likely that ER here represents the estrogen receptor. And so we can apply these at scale to correct for mistakes that the various input systems to INDRA make. There are several other error correction processes that I won't go into detail about now. So now we also have to deal with standardizing concepts across different nomenclatures and ontologies. Here's one example. So we have a representation of prostaglandin E2 in several different ontologies like CABI, MASH, drug bank. These look like different entries, but they actually represent the same thing. And so somehow we have to take these different groundings and create a canonical representation of prostaglandin E2 that maps across all of these resources and resolves this kind of redundancy here. So our solution to that is to have an ontology graph built into INDRA that draws on many different namespaces and taxonomies, CABI, MASH, GO, and so on, and imports mappings across these resources to be able to standardize concepts. Here again, the example with prostaglandin E2 that appears in all of these namespaces under different identifiers, and this is a subgraph of the INDRA ontology graph where the edges represent cross-reference mappings between these, and this is how we can actually end up with a standardized representation of a concept like this. This is extremely important. It's a huge problem that even the different human curated databases, but also some of these automated extraction systems, they just provide identifiers for the same concept in completely different namespaces that one has to somehow resolve. Now going to the, and as a side note, we started the biomappings project, having recognized that many of these cross-references that this graph is built from are actually missing between primary resources. We created a web-based curation workflow that can be filled with automatically proposed potential cross-references between namespaces and then curated through an intuitive interface on the web. The data being made available using a permissive CC0 license on GitHub so that it can be imported into different applications and propagated back into the primary resources. And this is one way in which we can fill in we can fill in gaps in the set of cross-references available among resources and then use them in INDRA. So this is the biomappings project that is available independently on GitHub. Anyway, so moving on to the statement level beyond entities, what we actually find is that a lot of the statements, like this example of RPS 6Ka1 phosphorylating CRAB1, are available at different levels of detail, different levels of abstraction. So all of these different boxes each represent an INDRA statement, here shown in English language for simplicity. that were actually collected from a given text corpus in a set of pathway databases by INDRA. And so on the bottom, you can see a very generic statement that a CRAB can be phosphorylated with 4 different text mining systems, having extracted more than 2,000 evidences for this pretty generic statement. And then as you go up this graph, you can see more and more detailed representations of the same process. For instance, CRAB is phosphorylated on a specific site. RSK phosphorylates CRAB on this specific site. A specific instance of RSK, P90 RSK, phosphorylates CRAB on this site, and so on and so forth. One more interesting thing about this is that the red dots here, they represent evidence from text mining. The blue dots represent evidence from pathway databases. So you can see that the placement of these is not completely random. So you can see that text mining tends to produce a lot of less detailed evidence for a given mechanism, whereas pathway databases tend to curate very, very specific context for these reactions, and there's a layer at which these overlap. But of course, it has to be understood that text mining also, while typically producing less detailed extractions, is also much broader. So it would produce extractions for a lot of things that aren't covered by pathway databases at all. In any case, another important task during knowledge assembly is assessing reliability and belief. What we found, you can see that on this plot here, which is actually a tabular representation of a Venn diagram, is that when processing a given corpus, we find quite a bit of complementarity across reading systems. These rows are each reading systems, and you can see that, for instance, this bar here is the set of extractions or set of INDRA statements supported by one reading system. Here's another one and the third one, and the ones where you see connections are ones where there's overlap, there is support from multiple systems for a given statement. So you can see quite a bit of complementarity across readers, which suggests that using multiple text mining systems allows you to extend the scope of what you can pick up. And yet, dealing with these overlaps allows you to also overlaps across readers and overlaps across multiple sentences and papers from one reading system allows you to assess your overall confidence in that extraction being correct, that is supported by the provided evidence. And so we built a probabilistic framework for calculating or predicting these belief scores associated with statements based on the overall evidence supporting them. You can, again, look at the refinement graph of statements and then calculate belief scores at each level and apply a cutoff to end up with the most specific and yet high belief statements that you can get, and ultimately use those for your modeling. You can see these highlighted in green in this particular example. And more generally, you can, through this approach, you can apply a lower belief cutoff to allow uncorroborated results into your model, or you can apply a high, more stringent cutoff, which would require having to have evidence from multiple sentences, multiple sources to back up the statements that you use. And this is an application-specific choice that you can make. And then finally, importantly, INDRA can also generate different models from the underlying knowledge. So on the left-hand side, you can see a representation of an INDRA statement. And in this example, we look specifically at rule-based dynamical models. And you can see how you can turn this phosphorylation statement into many different representations using a different policy in a rule-based framework. So for instance, you can use a one-step pseudo first-order representation of phosphorylation. You can use a Michaelis-Menten rate law. You can use a two-step policy where the kinase and the substrate first bind to each other to form a complex, and then the phosphorylated product is released. And in a more complicated setup, you can actually represent ATP binding to the kinase explicitly, for instance, if you want to then model an ATP competitive drug. And so this aspect of being able to use the same underlying knowledge to generate multiple model representations, this works at the level of rule-based dynamical modeling, but it's also more generally true in that you can generate causal networks and other types of representations from the same underlying knowledge, Boolean networks, and so on. Okay, so that's the basic operation of machine-assisted modeling. Here are some applications that we used this technology on. So we built a framework for natural language modeling where a human expert can describe a mechanism as a set of English language sentences, which gets processed and turned into INDRA statements and then generated into a rule-based dynamical model that can be simulated. And then the human user can then look at the properties of the behavior of this model and potentially tweak the sentences to change their hypothesis to adjust the model's behavior until they end up with a hypothesis that corresponds to what they expected. We also performed large-scale knowledge assembly at a literature scale where we took all available publications in PubMed, PubMed Central, and so on, processed them with multiple text mining systems, took inputs from pathway databases and created a queryable database of this, and then extending on the causal mechanisms represented in this database, created a graph database, a knowledge graph that extends on this using relations representing data and static properties and taxonomy. And I will talk about this in a little bit more detail. We also implemented a network search tool that can find causal paths in this knowledge. We've used our automatically assembled models to systematically explain cancer gene dependencies and also drug perturbations and their observed outcomes. We used automated modeling both to draw on this automatically assembled knowledge and to evolve and represent models during human-machine dialogue, like you see on the image here. You can build community-specific data portals based on this assembled knowledge. For instance, a knowledge base about understudied kinases derived from pathway databases and literature. We implemented a framework for keeping track of a set of self-updating disease models, where each model has a well-defined scope and reads a subset of the daily literature to update itself. You can also use this kind of automatically assembled knowledge for network representation learning with applications, for instance, in context-specific gene function prediction. And in another project, we also generalized the same approach to other types of systems beyond biology, namely to model socioeconomic systems. And there's a separate repository called INDRA World where an instance of INDRA is available for this generalized use case. And so for the remaining time, I'm going to talk about some of these applications. So again, this INDRA database, the idea behind this is to create one canonical assembly of all the knowledge we have available for processing and make it queryable through a web-based portal and a REST API. So to try to describe the scale of this, there's on the order of 25 million abstracts available in PubMed and 3 million full text articles available. Again, about 4,000 new papers are being processed each day. And we process on a daily basis all this content with multiple text mining systems. And we've also imported content from various pathway databases to end up, and these numbers are constantly changing, but roughly we have on the order of 12 million unique biological mechanisms with 38 million pieces of evidence from the literature supporting them. And you can actually query into this database. And here I'm showing these English language questions, but they can be done, and this can be done in a programmatic way as well. So for instance, what does CDK12 phosphorylate? What activates SHIP2? How does JAK1 affect STAT3? And depending on how you're doing this query, you can get like a browsable web page response where you can link out to different pieces of evidence, or you can get a programmatic representation of the statements that constitute the response. And you can interact with the INDRA database at db.indra.bio, and there's also a REST API with documentation available on GitHub. Going beyond this, so again, the INDRA database, it's somewhat limited to things that can be represented as INDRA statements that are mostly causal mechanisms. But when we do discovery or hypothesis generation in biomedicine, we very often draw not just on the underlying biochemical mechanisms, but the surrounding data and the static properties and taxonomy. And so we actually expanded on these causal mechanisms in a graph database, a knowledge graph representation framework implemented in the Neo4j graph database as a Neo4j graph database that takes everything we have from INDRA and extends it with relations representing ontological relationships, things like Go annotations, expression of proteins in different tissues, mutation profiles in different types of cancer cell lines, clinical trials data, and so on, to end up with a graph on the scale of 3 million nodes and 18 million relations. This is one example, and we call this the INDRA context graph extension. So this is one example of how you can answer a question like this from this graph database. So for instance, what we can ask, what drugs target calcium channels that are expressed in dorsal root ganglion? So the drug target information, those are parts of causal mechanisms that can be represented as INDRA statements, but the membership of specific genes in the calcium channel family, as well as the expression of proteins in a given tissue, those are things that are data and taxonomical relationships. And the color coding of these three types of relations, you can also see here in this subgraph. That was actually created by querying the graph database using the Cypher query language. And then you can see how you start from calcium channels. You can traverse the family relationship to get to family relationships to get to the level of specific genes. You can then look at the expression of those specific genes in this tissue, DRGs, and then you can look at drugs targeting these proteins. So we are also really interested in trying to be able to query this by its scale interactable knowledge using human-machine dialogue, which is a lot more intuitive than writing these graph queries. And our approach to that is actually to try to break up complicated queries into linked, simple question answering instances in a kind of sequence. So the same query as the one here, you can actually get the answer to that in three much simpler but linked questions. So you can first ask, what are the members of calcium channels, to get a list of genes that are considered calcium channels. Are any of those expressed in dorsal root ganglion? Here you are referring back to the previous result using a coreference and get a response based on that these are the specific genes that are expressed in DRGs. And then you can ask, are there any drugs targeting any of those, to get some drugs that target some of these proteins. This is actually an existing Slack application, this human-machine dialogue system called Claire, that you can integrate into your Slack workspace, and it can maintain different dialogue sessions for different either private messages or channels, each with their specific context, because of course you need to maintain context to be able to answer these interlinked questions. And one example of an integration for this Slack bot is in the COVID-19 disease map community, where over the months through which the COVID-19 disease map was created, dozens of curators interacted overall hundreds of times with the Slack bot to ask about various mechanisms relevant for curating the disease map. So shifting gears a little bit, I'm going to talk about these self-updating models that can keep track of discoveries in a given subfield of biology. And looking at the example of COVID-19, which is very interesting since it's a subset of literature that has very rapidly suddenly appeared, and then very rapidly evolved over the last couple of years. Here is a plot of the number of publications about COVID-19 in PubMed starting in January 2020. You see that for a couple of months, there's a small number of publications that appeared, but then suddenly starting in March 2020, a constant stream of about 300 new papers a day about COVID-19 started appearing. And this trend is still ongoing. And now there's about 200K papers about COVID-19 specifically, not to mention all the literature that is relevant for understanding COVID-19 from prior science. Interestingly, the composition of the COVID-19 literature is also shifting over time. Just to highlight a couple of examples, here's an early peak in hydroxychloroquine-related publications, and then much later ramping up kind of publications about COVID-19 vaccination started appearing, and now they are much more prevalent in this subset of literature. So we created a framework called EMMA, the Ecosystem of Machine-Maintained Models with Automated Analysis, that contains multiple disease and pathway-specific models, including one for COVID-19. And these models, they evolve over time with new discoveries. That is, they process literature every single day and extract statements from literature and extend themselves with new knowledge. They are running in the cloud autonomously. This is a graph of the number of statements in a given model over time. And importantly, these models can also be applied to try to explain key experimental findings. And interestingly, as the model evolves and as the model collects more and more knowledge, over time, it can explain new observations that it wasn't able to explain before. To explain this through the update cycle of the EMMA COVID-19 model, again, each day we extract new knowledge as soon as it appears from, in this case, from about 300 new COVID-19 related publications. These are assembled into the model, and the model actually tweets. You can follow it on Twitter. It tweets about what it has learned on a given day and links to the portal where you can browse the new statements each day. Then it runs relevant analysis, and I will show some examples of what that looks like on this model to explain certain observations, notifies users about new analysis results, again, also generating a tweet, and makes available these models through the EMMA portal for scientists and clinicians to register scientific questions and then get notifications by email if there are any meaningful updates to their scientific questions, given a daily model update cycle. More generally, we believe that this constitutes a novel way to interact with the evolving scientific literature. Instead of manually searching PubMed or signing up for keyword-based PubMed notifications, this takes a view of knowledge in the scientific literature through modeling, where you not only where you actually have a principled way of evaluating how new knowledge published affects a model's behavior. So to put a little bit more detail into what these models look like that we use to try to explain experimental data in this EMMA context, we can generate models at different levels of abstraction. So we can generate simple directed protein interaction graphs or logical models or rule-based models and their influence maps, and at the extreme chemical reaction networks that can actually be simulated as ODE models, for instance. On the left-hand side, these types of models tend to scale much better to large amounts of knowledge, but miss a lot of important causal context. So for instance, context represented by post-translational modification states, whereas chemical reaction networks have the potential to represent very detailed biochemical context, but typically don't scale to very large models. So our approach is to generate several different types of models, ranging from detailed to less detailed, and then try to answer the same set of questions using different models, and then you get different results and can learn from each of these levels of abstraction. Here is one specific example. We actually collected thousands of these drug-virus relations, like some where it was observed that the drug proceleridin inhibits SARS-CoV-2 replication. And using the EMMA COVID-19 model, we can automatically reconstruct a hypothesis for what a mechanistic path might be that could explain how this drug inhibits SARS-CoV-2 replication. And this is the hypothesis that EMMA came up with, that is proceleridin activates AMPK, which in turn activates NFE2L2, and NFE2L2 can inhibit SARS-CoV-2 replication. Each of the steps in this path are supported by literature evidence. So for instance, here NRF2, which is another name for NF-E2L2, can be modulated to reduce the potential of SARS-CoV-2. This is the one underlying evidence for this relationship. And then you can, of course, trace these to source publications. So for instance, here's a paper that hypothesizes NRF2 as a potential strategy against COVID-19. As I mentioned, users can also register queries with EMA. So for instance, as a very simple query, you can ask what inhibits COVID-19 and register that as a query once you log in. And from then on, you will receive daily email updates with any novel findings about hypothesis for how you can inhibit COVID-19. One example is this one. We hypothesize that pioglitazone can activate ACE2, which is known to drive COVID-19, the COVID-19 disease process. And here's an underlying publication. Can pioglitazone be potentially useful therapeutically in treating patients with COVID-19, right? And finally, we also integrated our human-machine dialogue system with EMA, so you can take a given EMA model, in this case, again, the COVID-19 model, and interact with it through natural language dialogue directly on the website. So you can ask things like what regulates ACE2 and get both a natural language response and a browsable site you can link out to. Do any of those affect TIMPRS2, and which of those are small molecules, to get a list of small molecules that regulate both ACE2 and TIMPRS2. And for instance, chemostat is one of the compounds that has been investigated against COVID-19. So you can relatively rapidly end up with answers that can help you plan your experiment or generate hypotheses. So with that, I'd like to thank our team here at the LSP, John Bachman, with whom we've led this team for many years, and he's contributed to many of the ideas and frameworks here, and our scientific software developers who took part in implementing a lot of these ideas. And I'd like to thank our collaborators from different institutions, for instance, those who develop text mining systems that are integrated with INDRA. I also would like to acknowledge DARPA for funding this work through several different programs over the years. So with that, I'm very happy to, one more thing. If you'd like to learn more about our projects, go to indralab.github.io. You can link out to all of the repositories and applications that I mentioned through this website. And we are also on Twitter. You can follow INDRA SysBio to get updates about our work. So with that, I'm very happy to take questions.
Thank you very much, Ben, for your very interesting presentation of this whole ecosystem of human-machine dialogue. So I don't see questions in the Q&A. I will take advantage of my role as a host, and I will shoot the first. So you said that up to now, for large-scale datasets, people mostly rely on statistical treatment and regression models. And while this is very informative, it kind of lacks the mechanistic explanation. So how do you feel we can reinforce this notion of not only relying on this type of networks that are mostly based on interactions and they have nothing of causality or mechanism? How would you, what would you propose to do in order to make this more popular and take advantage of this big omic data era that we are living in?
Mm-hmm. Yeah, so again, I think that the key is to try to breach the scales, to try to be able to, using machine assistants, build larger models with more mechanistic detail, and then use those models. You can conceptualize these as prior networks, or you can conceptualize these in different ways, but in some shape or form, take them into account when performing statistical analysis, enrichment analysis, and so on. There are many interesting lines of work, for instance, from the Saez Rodriguez lab, to try to draw on large-scale prior networks to interpret high-throughput data. All of these are attempts to try to bridge this gap. And I think that text mining specifically, first of all, it has the potential to extend on knowledge, especially rapidly evolving knowledge where the knowledge hasn't yet been curated into canonical databases. And it also has this potential of really enriching our interactomes with underlying evidence, linking to underlying evidence. So specific sentences that describe a given relationship. So even if it weren't to necessarily contribute anything novel, it still enriches all our findings by linking it back to the primary literature.
My next question is about this. So can we filter what we retrieve from the literature? Because many times what we can retrieve are fragmented information that might come from different experimental settings, from different experimental systems, and we tend to bring them all together. So many people argue that this can introduce a lot of noise also. And how can we go about it? What can we do to limit the false positives that we introduce in this type of prior knowledge networks?
Yeah, it's a very good question. One of the most interesting questions about this whole process, I guess. So first of all, there's a distinction between technical sources of error and kind of scientific considerations. So technical sources of error are things like text mining errors. When you read a given sentence, the text mining system makes a mistake. We can actually try to account for technical sources of error by just processing more text, using more systems, trying to align them. And in that sense, the scientific status of a given statement plays fairly little role, as long as the sentence describes an interaction that you want to reconstruct. It can help in eliminating technical sources of error. When we get to the scientific considerations, we can think about context, like what experimental context or what biological context was the result reported in. And to some extent, these machine reading systems can read for context, so they can recognize tissues, cell lines. If a given sentence mentions an observation in a cell line, the text mining system might be able to annotate that. This is kind of spotty and not super reliable, but there's some of this being done, and it's definitely something that is to some extent available. Another very important tool is human curated annotations directly on the literature. So if you look at PubMed, the Medline subset of PubMed actually contains annotations to medical subject headings for every paper that they curate, which is actually a very large subset of articles cataloged in PubMed. And those annotations can help you triage your papers into different categories. Like this paper is about cancer, this paper uses a mouse model, this paper talks about a clinical trial. So those kinds of annotations can also really help figure out the scientific status of or the context of a reported finding. And in fact, in many of our applications, I didn't talk about this, but in many of our applications, you can query for INDRA statements that were reported in a specific context, like cancer versus diabetes.
I'm going to profit from your last sentence and jump to the first question in the Q&A from Dr. Murray. For the text mining aspect, I may have simply missed it, and if so, I apologize, but how do you account for typos or other forms of human error that may skew the data set? Do such an error stand out, or is it a subtle impact at the final model?
Yeah, so typos are especially, not just typos, but just variations and diversity is a big challenge, especially when recognizing named entities, that is names of proteins, names of chemicals. It's a very big challenge. It's one of the most difficult things when adapting text mining to biology. And generally speaking, the way to solve that is to, on one hand, build large dictionaries of names and synonyms from resources. So if you look at UniProt, it lists a number of synonyms for every protein. So you can build these large tables to start with. And then on top of that, you can implement approximate string matching that can account for variations in capitalization, dashes, spaces, Greek letters versus spelled out forms, Roman numerals versus Arabic numbers, all these variations, plus things like typos, for instance. Some approximate string matching can account for those. And then on top of that, you often need disambiguation. I gave that example with ER, where it's not enough to just recognize the string and match it to some entries, but you also have to figure out, based on the specific context, which sense is most likely. There are other kinds of typos and curation mistakes, sometimes not even not completely random. So for instance, when you mention amino acid sites in pathway databases or in text, different authors use different conventions for numbering those sites. Some of them provide numbers of human proteins, others mouse proteins or rat proteins. You can either take the initial methionine amino acid into account when numbering or assume that it's cleaved off. You can take a signal peptide attached version of the protein or the signal peptide cleaved version of the protein and end up with different amino acid sites. And one of our side projects actually called the Prot Mapper is actually aimed at addressing this specifically, like accounting for mistakes and different conventions in annotating amino acid sites. So there are many, many such sources of error and ambiguity that one has to deal with.
Yeah, I will ask the second question in the Q&A. So it comes from Dr. Vera Licona. This tool is super interesting. I agree. I haven't used the tool yet. It was not clear whether you track the context of the curated interactions. Even if you don't have the info for every interaction, to be able to capture that info would be very useful. So do you track curated info?
Well, yeah, so again, to some extent, the text mining systems capture or extract context, and we do keep track of those. And second, because all of these evidences are grounded into the scientific literature, we can bring in these medical subject headings annotations that PubMed provides to track context, and you can actually query with context in mind when trying to find INDRA statements, for instance, through the INDRA database. So that's how we keep track of context.
To elaborate a little bit on this, text mining can have high false positives because sometimes the words are close in the sentence, but you know that the human language is very rich and it's not very easy to capture everything. So sometimes a causal interaction might be there when it shouldn't. So have you performed any tests with human curation of the same data set of, 10, 20 publications and the text mining? And do these compete or are the false positives very high? So do we need to redo the human curation once we have done the machine assembly, or we can be relaxed that it's as reliable as possible?
That's a great question, very interesting topic. So yes, text mining is highly error prone, and false positives is one way of thinking about it, but just mistakes more generally. It makes mistakes. We've done extensive work on trying to evaluate individual reading systems and this idea of combining across sentences and papers and reading systems and then calculating these belief scores. I myself have curated more than 6,000 sentences for correctness, and overall we have a corpus of more than 10,000 curations that we can use as a training set or as an evaluation set. And what we learned is that when you have a single evidence from a single reading system supporting a statement, there are differences between the specifics, but basically the precision is like 50, 60, 70% in that range, right? However, when you start seeing evidence from multiple reading systems, from multiple sentences, then this precision gets higher. And you can actually, especially from this training data, you can derive calibrated models of that can predict the correctness of a given statement based on the overall evidence. And we do find that we can set kind of a threshold that allows you to get to a better place in the precision recall curve. So you can't fully eliminate errors, and precision always comes at the price of recall, and that fundamental trade-off is still there, but at least you have a way of rationally making a decision for what to include and what to exclude based on your error preference. So some analytical methods are not that sensitive to errors, whereas things like detailed dynamical modeling can be very sensitive to errors, and so depending on your application, you can choose.
So how can one implement this ecosystem to its work? First of all, is it open source? Is the code available online? Do we need to install certain things? If someone would really like to give it a try, where should one start?
Yeah, so I think that the website I mentioned, indralab.github.io, is the best place to start, because that's like a single landing page that links out to all our repositories and applications. We are everything we do is open source and on GitHub, not just upon publication, but through its entire life cycle. So we work constantly publicly on GitHub, and all our issues and pull requests and all that is all public. I think that's actually extremely important, and I hope that this approach can be more widely adopted in the community, because it's actually a big problem also in BioNLP specifically, that a lot of systems are just not really available. They are published as methods but not made available. In any case, so yes, all of them are public, and it depends on your usage. So you might, looking at INDRA, for instance, you might want to use it directly as a Python package and implement your own programmatic workflows for knowledge curation and assembly. Or you can talk to one of our web services like db.indra.bio or one of the other services, either interactively or programmatically, to make use of it. And so you can also reach out to me directly with questions, and I'm happy to follow up and help.
And are the formats standardized? Does INDRA ecosystem support SBML or SBGN, some of the systems biology community standards, either for input or for output of the work?
Yeah, definitely. So, for instance, as input, you can INDRA can take BioPAC's input and derive INDRA statements from BioPAC. So that would be one example of a community standard. As for output, yes. So SBML, SBGN are both supported. There's one caveat though, which is that if you look at SBML or SBGN PD, for instance, it has this kind of reaction semantics that can have an aspect of combinatorial complexity when you generate a model. So INDRA statements, I didn't go into this in detail, but INDRA statements are more naturally aligned with rule-based modeling. And when you go from a rule-based model to a reaction model, you can end up with a huge number of reactions to represent the underlying complexity. And some of these export modalities like SBML and SBGN can suffer from this issue. And so that's one limitation, but yes, they are available as export formats.
But these are also challenges, not only limitations, but challenges to deal with in the future if people are interested in incorporating and implementing this approach into their research.